Our next speaker is Mark Adams, who is a research fellow in the Edinburgh team. And Mark is going to tell us about expanding the sample size and representativeness of genetic datasets through novel recruitment approaches. Thanks, Mark.
Thanks, Judith. So there are a few challenges that we have for understanding the biology and targeting treatments in mental health disorders. So the first one is just discovery. What are we looking for? And the second challenge is representativeness. Who are we studying to answer these questions? So the turn for the first major topic, discovery, this is just a chart showing the growth in genome-wide association studies for 3 major mental health disorders: bipolar disorder, major depressive disorder, and schizophrenia. And you can see from this chart that, sort of the last 10 years, there's been just a huge change in our knowledge on the molecular level of these disorders. So for example, in major depressive disorder in green, as recently as 2012, 2013, we sort of had 0 or maybe 2 parts of the genome that were reliably associated with major depressive disorder. And now in the most recent paper, we're finding over 100 variants associated. And then the sample size has also been growing, sort of exponentially. We're looking at the next wave of these studies will have well over 1 million individuals in them. But we sort of know, we're still finding just a very small portion of the signal here. And to truly capture all of the genetic architecture of these disorders, there's some theoretical analysis showing that we might need something like 10 million cases to really nail this down. So there's a need to get even larger samples than what's available now. So the second problem is representativeness. So I've looked at this in UK Biobank just as an example. And the problem with these kind of samples, as Andrew was saying, is they tend to be, sort of restricted ethnicity, better educated, and healthier. So what I did is a genome-wide association study of 2 factors that go into whether we have data on certain kinds of people in UK Biobank. The first one was just simply whether they had registered an email address with UK Biobank to make them recontactable for follow-up. And the second was there was an online survey of mental health, and we looked at whether people who were invited to this survey participated or did not participate. So there's this kind of layers and layers of filtering that's going on here. First, you've got to be recruited in UK Biobank, then you've got to be available for recontact, and then you've got to, once you receive the invitation, actually take part. So I ran these 2 genome-wide association studies, and then I correlated that signal with the genetics of a whole bunch of other traits. So health factors, mental health, behavior, body shape, just kind of the broad spectrum of things here. And we can see from these genetic correlations in red that are negative that people who provided an email contact or who took part in this mental health survey tend to have genes that would increase, make them healthier, less prone to disease. And this was also true for mental health traits. So people who took part that we have more detailed mental health information on actually are depleted for genetic variants that increase their risk for mental health disorders. So to sort of start to see like how can we expand the kind of data we have. We've been doing a little bit of linkage studies with trying to combine the genetic data we already have available with health records. So one way we've done this is in the Genome-wide Association meta-analysis of antidepressant treatment response. So the first sample we used is our Generation Scotland sample, where we had prescribing records from NHS Scotland, and we could just see whether people had been on one antidepressant, switched to a second type, and then switched on to a third type, is an indication that they, they were not responding to the treatment. And then we conducted a meta-analysis with the GenDep sample, which is a clinical study of antidepressant treatment response. And from this, we found that antidepressant treatment response had a shared genetic architecture with major depressive disorder. So because Generation Scotland is a family sample, we were able to break up this signal into sort of 2 portions, one from common variants that are shared by distantly related people, and also from rare variants that are shared by close kin. And we also found that this antidepressant non-response was also positively correlated with neuroticism, mood disorder questionnaire, which is sort of a measure of bipolar disorder, and then just general health questionnaire as well. So this sort of shows, it is possible to combine these two types of information. So now what we want to do in Pathfinder is sort of try to expand this into larger samples. So in Scotland, there are two efforts underway currently. SHARE, which is a linkage sample of people in the NHS, and also the dry Guthrie spots. So SHARE is a health register of older individuals in Scotland. It's people who they've consented to have their NHS records linked to data, and then they become eligible to participate in research. And then what happens is whenever they come in for any kind of care and they have blood drawn, there's the option for them to consent to their blood being diverted for genotyping and then made available for research. So within SHARE, we've done a pilot study to see if we can get more detailed mental health information from them. So we've conducted an online pilot, and the survey we're putting out just has demographic information and then various measures of mental health, substance use, and then some sort of life history, difficult experiences information. The completion time, as far as how long it take to complete the survey, we're aiming for sort of 20 to 25 minutes. And in the pilot study, we found that this was actually what we observed. And no one really spent more than an hour completing the survey. So important for the record linkage for these people who followed the survey were able to go in and access their NHS records to get more information about them. But importantly, their survey answers are not linked back to their NHS records. And then all of this data is stored on the data safe haven. So now what we're looking at doing is doing a genetic study with sort of two types of information. So we have people's health records from the NHS. So this is potentially, ideally, all of Scotland. So it would be a large sample, highly representative because we're capturing everyone, but the phenotyping is more minimal. So we know someone had a diagnosis of depression, but we don't have any additional information about exactly what their symptoms were or what life experiences they've been going through that could have led to increasing their risk for developing depression. On the other side, we have the online survey that we're conducting, which is sort of more targeted, a lot more specific information, but it has that same problem of people are sort of having to select themselves into taking it. So there's the problem of representativeness. So also just within the Pathfinder, we're sort of helping to provide infrastructure for both of these components, that we've got some freezers and genotyping for NHS Lothian. And for the full sample that we've rolled out based on the pilot survey, we're up to about 9,000 people who have responded. So we're in the process of accessing their genotypes and records for further investigation. So what we're looking at doing, sort of once we have the genome-wide association studies of these two things available, so the GWAS from the health records and from the online survey, is to find a way to combine it with GWAS from clinical cohorts, such as the ones from the Psychiatric Genomics Consortium. And we're looking at conducting a factorized GWAS where from these three cohorts, we can pull out, well, what genetic signal is common among all of them? And then after you've done that, you can sort of separate out any kind of genetic signal that would be specific to the SHARE sample, say because they're Scottish. And then finally, we would have an online factor that would represent what makes the people who are non-representative and fill out the survey different in their mental health genetics from the rest of these samples. And therefore, we can conduct an analysis where we're using all of the information that we have, but we're modeling out any of the effect of non-representativeness in the more complete online survey. And then once you've done that, you can sort of look at across the whole genome, which variants are just related to the disorder, the common disorder factor. So then I'll just finally finish up by saying briefly about the Guthrie blood spots. So what these are is, it's a test for inborn error of metabolism that's conducted in newborn infants. And these dried blood spots are put on cards for preservation and stored by the NHS. Currently, there's a research moratorium on accessing this data, but we've conducted a few feasibility studies. First, just showing that you can get genetic information out of these dried blood spots, even though they may be decades old. And also it's possible to link these to research cohorts like GenRich in Scotland. And also within Pathfinder, we've conducted some surveys of the public and find that there's a lot of support for researchers using and accessing this kind of data. So I'll finish there and just thank everyone in the Edinburgh team and in Share for making this all possible.